morning, everyone. It's a pleasure for me to participate in this summit. It's been a while since I've participated in a conference, so I am actually quite excited to be here. This morning, I'm going to talk to you about some innovative trial designs. I think it ties in nicely with Dr. Lawrence's comments on randomized controlled trials and the need for ethical clinical trial designs. My objective is to encourage you to engage a statistician early on in a trial design process. A trial design statistician has a rather robust toolbox from which we can draw on different design options, but those options have to be considered fairly early. And we can leverage all available information to improve your design efficiency. My agenda is to introduce myself, but I'll forego that since it was just done for me. I thank you for that. I do want to touch on the clinical trial workflow from a statistician's perspective to help you understand how we go about the job of designing a clinical study and the role of the statistician in that process. We're going to take a slight detour to give you an overview of what I call Bayes thinking, because many of the innovative designs these days rely or draw upon Bayesian methods or Bayesian concepts. Some of the innovative designs we'll touch on are adaptive by design, baskets and umbrella designs, platform trials, and lastly, incorporating historical data, that is data outside your clinical study itself. So the study process, and this is from a statistician's perspective, this is the data flow that we all experience in a clinical study. We recruit patients, we record data about those patients, data management, CRA, site get busy cleaning the data. The database is locked, the data is handed over to a team of statisticians and programmers who analyze and summarize the data, and those results are passed off to the sponsor who writes a clinical study report or a publication or both. At the design stage, this is how we as statisticians think about the study data flow. We start with your objective. And when we say objective, what question do you wish to answer? Or even better, what statement do you want to write in your study report or your publication? From that objective and that statement, we identify endpoints and populations to study, and we come up with options on what analyses might be used to answer your research question and meet that objective. Once we know what analyses we might use, we consider the data requirements to support that analysis. For example, we might say the analysis is mixed models, repeated measures, so we need repeated measures. We need data to be collected at given time points for specific data fields. Once the data requirements have been identified, then we can consider the design options. As a consulting statistician, I like to present different options to my clients, and each has a pros and cons for us to consider. After the design options are narrowed down, it's only then that we should answer the question, how many patients do we need? That is the sample size question. Oftentimes I am asked this question at the very beginning before the rest of this process takes place. And I try to quickly go through this process in rapid fire to get the answer to a sample size question. But ideally, that is at the end. And our role as statisticians in this process is to be part of a team. If you've written protocol, started studies, you all know this, the different stakeholders that are involved, the investigators, doctors who know patient populations and the care of the patients. Regulatory affairs is a group that we as statisticians work very closely with when designing a study. The regulatory affairs experts, they lay out the strategic pathway to engage the regulatory authorities such as FDA, European Medicines Agency, Health Canada, so forth. And it is the statistician's job to take that pathway and make an efficient journey of it. We do speak with clinical operations about practicality of collecting data, product experts to try to understand the mechanism of action and so forth. So we are part of a much larger team. We cannot work in isolation. And our main job as statisticians during the design phase to make your study more efficient by helping it to run more quickly, finish sooner, with fewer patients. We do that by lowering the sample size, and that is done by reducing variability in the study population. Now let's look at this slide that shows three distributions. It says mean at the bottom. Since we're in a pandemic, think of length of stay as an endpoint of interest. We want to reduce length of stay. And that mean is the average length of stay for a patient of a given population. The green distribution shows that it's quite common and perhaps likely that a patient is going to deviate from that mean. With high probability, they might be above or below that mean length of stay in a hospital. In order to get a good estimate of that mean in this population, we need a high sample size. The blue line shows a population where they are less likely to deviate from that average length of stay. There's more information in the same number of patients, or you could get the same information with fewer patients, as we discussed with the green population. That's median variability, and of course, the red distribution would be ideal. We don't need a lot of patients to estimate average length of stay in this population because it's unlikely they're going to deviate from that average from our estimate. So we, as statisticians in the design of a study, are trying to reduce the variability at the level of randomization in a randomized trial so that we can get the information we need with a low sample size. Now, we are going to take a sidestep or a detour to discuss Bayesian thinking. Bayesian methods are used, I don't know about a lot, but frequently in some of the innovative designs, as we will see. And I've tried to discuss or describe or explain Bayesian statistics or Bayesian methods without much luck. So I think what's important is how do we think like Bayesians? What's the principle behind Bayesian methods? In brief, it's a way from learning from our experience. We have a prior belief about some event or some occurrence. We have a new experience, we collect new data, and we change our prior belief. We update it to something more reasonable based on that new experience. And that's the principle behind Bayesian thinking. The example on the screen is very, very simple. Focus on the POP, the POP, probability of precipitation. When we get a weather forecast, each of us interprets that probability differently. 30%, we might not take an umbrella with us. We don't think there's much chance we're going to get wet. But if POP rises to 50%, maybe more of us will grab that umbrella before we go out the door. Now let's suppose that it's a very rainy week or a week where we have POP of 50% for 5 consecutive days, 8 consecutive days. And on some of those days, we get wet because we never grabbed our umbrella. Well, that's new data. That's new information. And I am going to update my prior belief of what 50% means and say, okay, it's more likely it's going to rain. I will take my umbrella. And that's the basic principle behind Bayesian thinking, is to update our beliefs about the probability of some event with the new information. Now, Bayesian methods were designed first and have their roots in assigning probable cause, which we are very interested in. Does our drug cause the effect that we're looking for? Or adversely, does it cause the adverse event or side effect that we hope it does not cause? This brochure showing tuberculosis symptoms, we probably would not have much difficulty in coming up with estimates of the probability of having a symptom given that we know we have tuberculosis. That's probably been measured quite a bit from all tuberculosis patients. They're going to have weight loss, fever, night sweats, tiredness, loss of appetite, and an enduring cough. But if we have the symptoms, we don't see tuberculosis. If we see the symptoms, we've loss of appetite and we're tired, how likely is it that we have tuberculosis? That's a much more difficult question. There are many other causes of loss of appetite and tiredness. So maybe we have tuberculosis or not. And the Bayes methods are used to reverse the probabilities. Instead of probability of a symptom given tuberculosis, what's the probability of tuberculosis given the symptom? Because we only see that effect in the symptom, not the underlying cause. I'd like to think about and investigate the history of things when they're new to me. Bayesian isn't new to me. Goes all the way back to my days at North Carolina State. But for some historic perspective, this is Thomas Bayes on the right-hand side. He gets the methods named after him. He was a reverend in England, and back in the 1700s, instead of writing blogs or social media, folks would write pamphlets, and David Hume wrote a pamphlet stating that we cannot declare cause without observing the cause and effect relationship. So he's an empirist. He wants hard data, hard evidence before we can declare cause and effect. This made the folks in the religious circles uncomfortable because there was this principle of first cause, a deity or a god that would cause things to happen, like the sun rising every day. But we cannot observe a deity pushing the sun up into the sky every morning. So we can't declare that God is the cause of the sun rising because we can't see that happening according to David Hume. So what the Reverend Bayes did was, using mathematics and the new science of probabilities, he wanted to refute David Hume mathematically that it is possible. So he worked out his theorem that gave us the way to estimate the probability of a cause given only the effects being observed. He never published, but it was uncovered after his death and published, and he's given credit for the Bayes theorem and Bayesian methods. So what does this have to do with our work in clinical research? Again, let's pretend that the horizontal axis, the parameter, is our endpoint of interest, its length of stay in a hospital. We see three distributions for an average length of stay. Let's focus on the dark blue curve first. That's our current belief. We believe the average length of stay is represented by the peak of that solid blue curve, and the variability of patients for that length of stay is represented by the width of that curve. Now, how do we get that belief? That belief might come from data. We might have early phase study data where we can estimate this distribution of an endpoint of interest. We might have publications. We might have data from external studies, registry studies. We might not have any data. And that's what the circled subject matter expert, where they come into play. Bayesian methods can be used when there's no prior data existing. FDA hasn't taken to that method yet, but I'm thinking someday it will be used more if the survey of subject matter expertise, the way that expertise is quantified, becomes systematic, much like literature reviews are today. Then we can survey our subject matter experts and formulate this prior belief or current belief. We use that information to design a study, for example, a phase 2 study, and the results after that phase 2 study are represented by the distribution with the large dashed lines. So we have our prior belief, solid blue, current data, large dashed line. The Bayesian methods combine these two. And the idea is that we update the distribution of the solid blue by combining it with the data from our current study and estimate what's called the posterior, or this distribution represented by the smaller dashed line. And don't be too concerned about how these are combined. It's not always going to be right in the middle. If we don't have much confidence in our prior belief, we can give more weight to the data for our current study. There's a lot of details that the statistician will work out. Here's a real-life example for disease-free survival. The blue is the prior, the green is from the data that we collected in a current study, and the new estimate is the red curve. If we're not done, we're going to conduct new studies, then this posterior or new estimate, the red curve, then becomes the prior for the next study as we plan that next study. This method can be incorporated into what I call Bayesian system. It can be cyclical. So I said that the prior is updated, becomes a posterior, becomes our new estimate. That then becomes a prior in the next study, and you can cycle through this over and over again. In fact, these Bayesian systems are at the heart of artificial intelligence. They do learn over time, and this is how machines can learn with the new data coming in and updating operationally. In fact, all of us carry with us a Bayesian system every day, and we look at it and spend hours on this Bayesian system every day. And if you're not paying full attention to my lecture, you might be looking at it right now. It's your smartphone. And every smartphone includes Bayesian models to separate transmitted signal from noise so that you get clear reception with your phone. And the improvements from the third generation to 4G and 5G was in part an improvement in those Bayesian models in your phone. So now let's look at the actual designs. Now what we've done is look at the process statisticians use to go from a study objective to determining the analyses, the data, the design options. We took a little sidestep to talk about Bayesian methods, which rely on prior beliefs, prior information, and update that information with new study data to come up with a new estimate. Now in the designs, the next few slides, I'm going to describe a design that we worked on several years ago for a client in oncology. And the next couple slides show some of the features of that design. And I put our three variability examples in the upper right corner as a reminder that what we're trying to do is reduce variability. Now let's say for a given study, two baseline characteristics that correlate with the endpoint of interest are the stage of a disease, whether it's early or late, and whether the patient received prior therapy, yes or no. If those two baseline characteristics are correlated to the endpoint of interest, then we stratify randomization by those two characteristics. So you will be randomized to active versus control within each of the 4 cells defined by those two binary characteristics. When we do that, because randomization occurs at a smaller level rather than overall, the variability in the population when we do the analysis is going to be much lower. Another design feature is enrichment, and it's similar to stratification in that we're dividing up the patients at baseline based on some characteristic. And in this case, it's usually a biomarker. Is where enrichment designs first came about. And they're more common in the early phase studies because we're trying to achieve a very homogeneous population, think this red distribution, to really tease out the signal from variability or noise. So let's study a very homogeneous population first to understand if our product is safe and efficacious in that small group. So what we do with that biomarker is we ignore patients who don't have the biomarker of interest, or the reverse, depending on what we need to do, and then we study that homogeneous group. Biomarkers have at least two flavors or reasons for enriching a sample based on the biomarker. One reason might be that the biomarker indicates that the patients who have this biomarker are more likely to realize an event that we're trying to study. For example, disease progression. Another reason would be that the biomarker indicates that the patients will respond to the treatment that we are offering. So it's one of those two. In this case, you see enriched for responders. So whatever biomarker this is, those who have it are more likely to respond to our treatment than those who do not have it. That helps us in early phase studies with this homogeneous population. It makes clinical trials possible because we can afford to study a smaller sample size and get the information we need. After we discover that things are working and they're safe, then in the later phase, we might enlarge our sample back to the larger population. And finally, for this design that I'm leading up to, we used what's called adaptive randomization as another innovative design feature. This comes out of oncology research out of Texas. I'm trying to remember the cancer, the oncology group down there. It escapes me right now. Peter Thal was the first one to publish on this idea, adaptive randomization. The idea is that we're not just adapting features of our study, we're changing the probability used to assign a patient to a given treatment arm. And this goes back to Dr. Lawrence's discussions or comments about is it ethical to treat patients with experimental products that may not work or may be toxic. And in oncology, there's two things we're interested in. One is toxicity, one is efficacy. We want our products to be efficacious with low toxicity. And we might want to study the dose level to get an understanding of how that dose affects both of those features. And what Peter Thal called the combination of these two is utility. A product will have high utility if it has low toxicity and high efficacy. And this graphs, they look the same for toxicity, efficacy. This is just a made up example. They don't have to be. And a lot of this to start with comes from subject matter expertise. But for example, dose level 1 in toxicity, it's most likely to have very low toxicity, level 1 toxicity, and very unlikely that at a low dose, you're gonna see a high level of toxicity. But its efficacy is most likely going to be very low. And the probability that it has very high efficacy is very low. And it's the reverse for the high dose. It's more likely to be efficacious, but with high toxicity, and dose level 2 is somewhere in the middle. And what we do is, as we randomize patients, we get an early indication on a surrogate endpoint, 3 months, 6 months, on what's happening to patients for a given dose in toxicity and efficacy. That is, we collect the evidence, we update that evidence, and the next patient that comes in might be randomized with higher probability to say dose level 2 and lower to dose level 3 because it's too toxic. And putting all of this together, this is the design that we came up with for our client. The FDA said they were not satisfied that the proper dose had been selected, so we needed to study doses again. But the business investors, stakeholders wanted to move on to phase 3. So we came up with this design to satisfy the FDA's concern about selecting the right dose, but move very rapidly into phase 3 to satisfy business constraints. And this did go before the FDA, and they had no concerns about this design. They rather liked it. I will note here that we did require a minimum sample size at each dose before we triggered the adaptive randomization. And then as the patient's data came in at 3 months, we might lower the probability for a given dose based on it's underperforming, it's not effective, or it's too toxic. Okay, next, you may have heard the concepts of basket and umbrella studies. Again, these are originated in oncology. I do believe they have applicability elsewhere, but let's first look at a traditional design. We have 4 tumor types or 4 indications represented by the shapes, and the mutations underlying this tumor cells represented by color. And in traditional methods for each drug and each tumor type would get its own clinical study. So even if one of our clients had same drug, but they wanted to study 4 different cancers, and in traditional design, that means 4 different studies. A basket trial takes a different look at this. Let's say the one drug addresses a mutation represented by blue, and that mutation occurs in all 4 different types of cancers: lung cancer, breast cancer, bladder cancer, what have you. So instead of doing 4 different trials, let's do 1 trial with that one drug all at the same time. Now, the way this becomes more efficient is we change the research question. Again, this would be in early phase studies. Instead of asking, does this drug work on, say, lung cancer, we ask, does the drug work in at least one of these cancers? Changing that question in at least one of allows us to pool the data across the different tumor types and get more information for a smaller number of patients. Umbrella designs, instead of one drug going after multiple cancer types, it's multiple drugs after one cancer type. These are becoming more common in the large pharmas, where several pharmaceutical firms are all developing a drug to treat the same cancer type, even if they have different mechanisms of action. So they can share data across, especially with the standard of care arm, instead of each pharmaceutical firm enrolling patients with the same size standard of care arm, they all use the same standard of care arm. So it is more ethical in that fewer patients are getting just a standard of care arm that maybe we think we can improve upon. What I find challenging about this scenario, especially when each drug comes from a different company, is the contractual agreements that have to be hammered out beforehand in sharing the data. But it could be one company with different combinations of drug that they're trying, which are common these days. Okay, platform trials. These are more recent development. It's a way of rolling out the basket umbrella trials. First, looking at the traditional design, this is what might be called a drop the loser design. We have 2 standard of cares, and we add a treatment to the standard of care. So 4 arms, each standard of care alone, and then that standard of care plus the treatment. And we plan to conduct an interim analysis, and if a treatment arm or arms is underperforming, then we would drop those and continue only with the ones that are performing as we need them to. The one thing I want you to note here, because we will contrast this later, is that all of the arms of interest are initiated at the beginning of the study. So I call this starting big. Okay, confirm sample size. The last note here is that in this interim, not only might we drop the loser, but we might confirm the sample size to continue this standard of care A forward. That's a common adaptive feature, is to re-estimate the sample size. Now, a platform trial is different. Instead of starting big, it starts small. We write what's called a master protocol, and that master protocol covers all the features for any drug or treatment arm that's going to be considered under this master protocol. Such things as eligibility criteria, randomization features, common objectives, maybe the endpoints. All of these have to be common across all what are called appendices. Now, each drug or treatment combination that we want to study then becomes an appendix to the master protocol, and it's attached on there. Those appendices are protocol that lay out what's unique about that appendix, about the population being studied in that appendix, about the treatment that's being considered in this appendix, and so forth. And the way these might be rolled out is over time. And I find this particularly useful for a small company. Instead of start big, is to start small. And I've increased this now to 3 standard of cares, A, B, and C. And we might think that we have the best chance of showing efficacy and safety with standard of care A. So we begin our platform study with that. Starts in January, and we enroll both standard of care patients and treatment patients. We conduct an interim. Based on this interim, we might start a new appendix. This graphic shows standard of care B starting a couple months after standard of care A, and it has a small standard of care arm and its own treatment arm for treatment B. If the standard of care patient populations are congruent, then with each appendix having its small SOC arm, we can combine them. And when we conduct this analysis for treatment A, we're going to combine the standard of care arm B with standard of care arm A, so we have a larger standard of care arm. And that's one of the big benefits of platform trials, is being able to combine data, whether it's standard of care or treatment arms. Now, those populations have to line up. Another advantage is that standard of care might change over time. And because we're starting new standard of care arms later on, we can capture that and study those changes of standard of care or account for them. So when we do the interim analysis 2, maybe we don't use the combination of all 3 standard of care arms, but just B and C. It provides a lot of flexibility. And these appendices, if they are underperforming, they can drop out. You add new appendices and drop out old appendices as the data indicate. So it's maximum flexibility. We start small and expand as the evidence accumulates. The successful appendices can be expanded and go on to a phase 3 study. Bayesian methods come into play by combining the standard of care arms, that is, we borrow across the appendices to reduce variability, give us a larger sample size, can be done for treatment arms as well. And we're going to talk about this in a moment. Historic data can be used to reduce initial sample sizes. The WHO has wrote a master protocol for studies around COVID-19. I just thought I would point that out as an example. We've designed a couple of COVID studies here, and we followed that WHO master protocol quite closely, even though the sponsors were conducting their own studies, not jumping into the platform trial that the WHO was running. Okay, the last innovation that I'd like to talk about is using historic data. This is more recent. I find this quite exciting for a couple reasons. One, it's really Bayesian in its thinking and its methods, and it can really reduce sample size if done properly. The idea is that we have designed a study. This is the experimental block. And we might have a small control arm, but there might be historic data available that we can combine with that control arm to get a much larger control arm. This historic data might be from other studies. It might be from registry studies, observational studies. It might be from publications. Better is to have the patient level data, but okay is to have from a systematic literature review endpoint estimates and variability and so forth from the published literature. We're working on a design right now that we will put before Health Canada, where a single arm study is we're creating what's called a synthetic control arm, where it all comes from historic data. And we're about to go in front of Health Canada to defend that methodology. The important point here is in this slide, which I think is from a presenter from Eli Lilly. There was a conference in Washington, D.C. in March, covered complex innovative designs. And this slide comes from one of those presentations, and they call it congruency. The patients in the historic data should match what's in your study design control data. So they should be congruent. If they're not congruent, may not be able to use those historic data, but there are lots of smart statisticians working on methods to make that possible as well. And I think that is all I have. A reminder to engage a statistician early. We're all part of the team. We do want to help you as best as possible to design an efficient study. If you do have any follow-up questions, there's my email address. And with that, Sam, I'll give it back over to you.
Thank you very much, John. That was a very interesting talk. And I know a lot of people in our audience will be thinking closely, especially as they plan ahead in 2021. We do have a question for you, John. Mm-hmm. You mentioned the ethical advantages of adaptive randomization in oncology studies.
Yes.
Could you kindly also comment on ethical advantages of a Bayesian approach with respect to informed consents of subjects and other benefits for subject welfare?
A Bayesian method for informed consent.
Yeah. Can you also comment on ethical advantages of a Bayesian approach with respect to informed consent of subjects and other benefits for subject welfare? And that was asked by Dr. Jeffrey Lawrence. And Jeffrey Lawrence.
Okay. Yeah, Dr. Lawrence, if you can, what do you mean by Bayesians for informed consent?
Can you hear me?
Yes, I can hear you.
Yeah, so yeah, I mean, it's basically an informed consent. Is there anything that you can put in the informed consent to inform the patients who are not statisticians mm-hmm. To understand better and maybe maximize their welfare? In other words, what you mentioned this, I think, in the oncology situation.
Yes.
If you're going to reduce the risk of getting a toxic or a harmful kind of thing.
Yeah.
Are there ways of communicating that to patients? Number one, to inform them better and maybe to make them more likely to enroll?
I see. Okay, so I think it would be important if we're using that type of design, a Bayesian method or adaptive randomization, to do our best to make it known to the patients that we're doing everything we can to ensure that they are going to, I hesitate to say them specifically, but to ensure patients do not receive either toxic, highly toxic, or ineffective treatments. That the designs are intended to move patients very rapidly away from those types of treatments to the treatments that are that have high utility, both effective. So I think and that's the challenge, is to put it in plain language, which is what I've tried to do in this presentation.
Yeah, thanks. That's really interesting, because I remember the bad old days of doing oncology, dose pushing to dose limiting toxicity, and
yes.
So I think
yes.
this could have some advantages.
I agree. It's becoming quite common in some of the oncology studies.
Great, thank you.
Mm-hmm.
We have one more question from Dr. Allison Gage. Are synthetic controls mainly acceptable in rare diseases, or are there examples in more prevalent conditions?
It's the former, more in the rare rare diseases, hard to find populations. The one we're working on is, again, an oncology study where it was a single arm study. It wasn't meant to go before regulatory authorities, so we're kind of trying to adapt it for regulatory approval post hoc, if you will. And there aren't a lot of patients with this indication. So rather than recollect information, recollect it from patients, we don't want to have to do a new trial and have a control arm. Let's see if we can leverage data from what's been done in the past by others. That's the main intent, but it is more common in the harder to find populations. Yes.
Wonderful. And yeah, if anybody has any more questions with John or wishes to connect with him, you can use our meeting scheduler and send him a request, or you can email myself or Sherry and my team. He's always available to connect with you. He's based in Toronto, and he's available as early as next week if you have any questions for data management or statistics. So thank you again, John.
Thank you, Sam.